Okay, so welcome back to this next video in which we are discussing cystic fibrosis and cystic fibrosis medications. Okay, right. So, we have just discussed the pathology associated with the pancreas. What we're now going to move on to is the pathology associated with the intestine and then the pathology associated with the male reproductive tract. Okay, right. So let's firstly go on to the intestine. Okay, so once again, we need a bit of anatomy and physiology. Okay, so we're doing the intestine now. Okay, so I'm going to show you what the wall of the small intestine looks like. Okay, so we're specifically going to look at the small intestine rather than the colon. Okay, so I'm going to have taken a little section of the wall, so let me just show you this. So firstly I'll just show you a tube which can represent the intestine. Okay, right, so what I'm going to do is I'm going to take a cross-section, okay, like we did for the bronchus, but I'm not going to draw the entire thing out, okay, so I'm going to chop through the intestine, okay, and I'm just going to take a little section. So if I got my cross-section, it would look like this. What I'm going to do is just take a little section of it and look at that portion, okay? So we're going to look at a little portion of the wall of the intestine, basically, and we're only going to look at the most apical layers. Okay, right, so this is what it's going to look like. You're going to have villi extending out like so. Okay, and then in between villi you're going to have little crypts that invaginate inwards, like so. Okay, so here's another villus, here is another one of these crypts. And then here's the bottom of the wall of the intestine here. Okay, so we're going to only look at the most outer layer of the intestine because this is the only layer we're actually going to need in order to understand this. So we're just going to have a look at these epithelial cells which line the intestine. Okay, so I'm just giving them a thickness now. So, let me get my highlighters. Underneath the epithelial cells then, which I'm going to show in turquoise here, you're going to have a basement membrane. Okay, like so, which is what all of the epithelial cells are attached to and what they're sitting on, and this is what's holding them up, basically. Okay, right. So I should probably split these epithelial cells apart, like so, okay, rather than having them all as one great confluent structure. Okay, so I'll just show it for a little portion rather than doing the whole thing. Okay, so we'll have this portion done properly here. Okay, so let's just label up some things here. So this is the basement membrane, which I'll just abbreviate down to BM. Okay, this structure here, which I'm going to box in orange. Okay, this invagination, this has a very fancy name. It's what's known as a crypt of Lieberkühn. Okay, so this is a crypt of Lieberkühn. Okay, then we have these evaginations. So the crypt of Lieberkühn is an invagination into the wall. Okay, then we've also got evaginations out from the wall, and this is what's known as a villus. Okay, now what I should also mention is that the epithelial cells that line the intestinal surface, these have a special name. Okay, they're known as enterocytes. Enteric means pertaining to the intestine. Okay, so enterocyte means cell of the intestine, basically. Okay, and these cells have little structures projecting off their apical surfaces that are very, very similar to the cilia, which project off the surface of the epithelial cells that line the bronchi. Okay, but these are not called cilia. Instead, they're called microvilli. Okay, and under a light microscope, when you view the wall of an intestine under a light microscope, you cannot make out the individual villi. Instead, what you see is a sort of hazy area above the epithelial cells. Okay, and this was known as the brush border. Okay, so the brush border is the name for that layer of microvilli projecting from the apical surfaces of the enterocytes. Okay, right, so what is the job of most of the enterocytes? Well, the job of most enterocytes is just to absorb nutrients, okay? So you've got a very good blood supply underneath the epithelium, which is there to take the nutrients from the enterocytes. You've also got, of course, lymphatic vessels, which will take certain nutrients, such as lipids, from the enterocytes, okay? Now, what we're going to be interested in is other functions besides absorption. Okay, so basically, the enterocytes are also coated in mucus, okay, and this mucus is going to help lubricate the food bolus, lubricate the surface of the intestine so that the food bolus can move easily through the intestine without getting stuck, basically. Okay, so you've got a layer of mucus over the surface of your epithelial cells of your intestine. Now, what is it that produces this mucus? Well, firstly, you're going to have goblet cells interdispersed among the enterocytes. Okay, so I'll put these in here. So those green cells, which will not have microvilli, those are goblet cells, okay? And we know by now what goblet cells do. They're going to secrete mucin, or mucin, that glycoprotein which is responsible for turning a serous secretion into a mucus secretion. Okay, it's the thing which makes the fluid viscous. Right, but what secretes the serous solution in the first place, okay, the water and the salt, which the mucin has to add into to become mucus? Well, it's believed that it's cells that are within the crypts of Lieberkühn which produce the serous secretion, which is going to mix with the mucin from the goblet cells to make the mucus. Okay, so let me put some of these on. So some of these special cells in the crypt of Lieberkühn, and I should just say, the crypt of Lieberkühns have other functions as well, okay? So the intestinal epithelium is not a fixed layer. You are not born with your intestinal epithelium and you keep it your entire life. Unlike your heart cells and your brain cells, which you do keep your entire life, the intestinal epithelium is continually being replaced. Okay, now at the base of the crypt of Lieberkühns, you have stem cells. So I'll just mark some of these in here. So they're not quite at the bottom, but they're pretty close to the bottom. Okay, so these blue cells here, this is just a bit of background knowledge, we're not going to need this. I'm just adding it to add a bit of extra stuff here. So this is an intestinal stem cell here. Okay, and these stem cells produce new enterocytes, which are continuously being given off, basically. And these enterocytes move up the crypts of Lieberkühn, the size of the crypts of Lieberkühn, and then up the villi, and then they get to the top of the villi, and then they're shedded off. So there is a continual cycle of these enterocytes, basically. They're continually being produced by the stem cells down here, then they make their way up, and then they're shed off. Okay, so that's another function of the crypt of Lieberkühn, that they're actually the source of all of the enterocytes. However, another function of the crypt of Lieberkühn is that some of these crypt cells here, shown here in red, are responsible for producing the serous secretion which the mucin is going to be added to produce mucus. Okay, so these are producing the serous secretion, which, remember, contains water and then sodium chloride as well. Okay, right. So, how do these crypt cells actually produce that serous secretion? Well, it's the same old pathway, the exact same story. So let me get it back again. So, it's this story that we've got here from the lungs. Okay, so once again, you've got your apical face of your epithelial cell, which this time will be facing into the lumen of the intestine. The basolateral face will be facing the basement membrane. Okay, again, you'll have sodium-potassium ATPases in the basolateral membrane, which will establish the sodium and potassium gradients across the cell membrane. The NKCC will bring in sodium, potassium, and 2 chloride anions into the cytoplasm of the cell, and it will use the sodium going down its electrochemical gradient to drive that movement. Okay, the potassium will then leave the cell for a potassium channel and then can continue to cycle. The sodium will be removed by the sodium-potassium ATPase, and the chloride anions are going to go out through CFTR anion channels, cystic fibrosis transmembrane conductance regulator anion channels, which are on the apical membrane and which will move the chloride anions into the apical extracellular fluid here. Okay, then of course what's happened is you've net moved chloride anions from the basolateral extracellular fluid to the apical extracellular fluid. Okay, that means that you've moved negative charge from the basolateral extracellular fluid to the apical extracellular fluid. So what will happen is sodium ions will move from the basolateral extracellular fluid to the apical extracellular fluid, just following the charge basically. And then of course you've moved solute, so water will then move by osmosis across the epithelium. Okay, and both the sodium and the water will go via the paracellular route. Okay, so it's the same mechanism again that underlies this serous secretion by these crypt cells. So, in cystic fibrosis, the cystic fibrosis transmembrane conductance regulator has lost function, so this serous secretion is hugely impaired. Okay, but of course the goblet cells are still going to secrete the same amount of mucin, so again you end up with a really thick, sticky mucus lining the surface of the intestine. And this is much worse at the lubrication of the surface of the intestine. So what ends up happening is you end up getting compaction basically. You end up getting obstructions in the intestine which are made up of fecal matter along with mucus that is really thick and sticky basically. Okay, so this is a big problem that people with cystic fibrosis face, that you can get compaction of the intestine in this way, which is going to obstruct contents moving through the intestine. And again, this is resulting because of the dysfunction of the cystic fibrosis transmembrane conductance regulator. Okay, right, so there we have the intestinal manifestations of this disease. What I now want to move on to is what's going to happen in the male reproductive tract. Okay, so the male reproductive tract. So basically, as I said at the start, what happens is you get congenital bilateral absence of the vas deferens. Okay, which is often abbreviated down to CBAVD. Okay, so the C is for congenital, the B is for bilateral, the I is for absence, and then there's of the, which don't get letters in the abbreviation, and then it's of the vas deferens. Okay, right, so in order to understand what this is firstly, and then to understand why loss of function mutations in the cystic fibrosis transmembrane conductance regulator are going to cause it, we need to firstly know something about the anatomy of the vas deferens. Okay, right. So let's start by drawing a testis, and then we'll draw another picture after that where we show the vas deferens joining with the seminal vesicle and then becoming the ejaculatory duct which feeds into the prostatic urethra. Okay, so we'll start by drawing a testicle then. So here we have a testicle here, and let's say it's the right-hand testicle. Okay. And basically, surrounding the testicle, what you have is a structure known as the epididymis, which I'm going to put here, which will become the vas deferens here. Okay, so the vas deferens is basically a tube that carries sperm from the testicle up to the ejaculatory duct, which then will join the urethra. Okay, right. So let me draw other structures on here. So there's also the epididymis. Now, what is the epididymis? The epididymis is a single tube, but it's not a simple linear tube. Instead, it's a tube that is coiled around and around and is incredibly convoluted. So I'm just thinking how I should show this. So even though the way I've just drawn it, it looks like a tube that's just a single linear tube. In reality, what's happening is it's convoluted around all over the place. But it is still a single tube, basically. It's just wrapped up and up and up and up. So this entire structure here is a huge convoluted tube that is in continuum with the vas deferens. So let's put some annotations on here. Okay, so this is the vas deferens here. Okay, and this structure then is the epididymis. Right, and there are different portions of the epididymis that are named. Okay, this portion that is up here, this is called the head of the epididymis. Okay, so I'll just highlight up these different portions. So this is the head of the epididymis here. Okay, then after the head of the epididymis, here in blue, you have the body of the epididymis. Okay, so the start of the great tube that is the epididymis is up here, and it is a huge great tube. I believe the estimated length of it is around 5 meters usually. Okay, so it has to be wrapped around a lot, basically. This portion in blue, this is the body of the epididymis, so further along this great tube. And then finally, right down near the point where you're actually going to become the vas deferens, here in red, this is what's known as the tail of the epididymis. Okay, right, so the head of the epididymis, the body of the epididymis, and the tail of the epididymis. Right, so that is a great convoluted tube. Now, let's talk about how sperm that are produced in the testicle are going to get into the epididymis. Okay, so another structure that we need to mark on is something known as the rete testis. Okay, so this is the rete testis. Okay, and I don't know if the origin of this being called the rete testis is the same as the origin of those rete ridges and rete pegs that we saw ages ago in the epidermis and the dermis. Okay, so what the rete testis is, however, is it is a very intertwined network of tubes. Okay, so if I show this, basically you've got loads of tubes that are connected to each other like so. Okay, so it's loads and loads of different tubes that are just all connected to each other. Okay, so it's a great meshwork of tubes. Okay, and what's going to happen is loads of other tubes that are in the main body of the testicle are going to empty into the tubes of the rete testis. Okay, so if I colour in the rete testis here in vivid purple, so this is the rete testis. The rest of the testicle is made up of loads of little tubes that are actually the places where sperm are being made, and these are going to be draining into the tubes of the rete testis. Okay, so the tubes that produce sperm are what are known as seminiferous tubules. So this is a that's not right. Seminiferous tubule. Seminiferous, I believe that's how you spell it, tubule. Okay, and in these tubes what will be happening is you'll be producing sperm, and these sperm will be moving up the seminiferous tubule up into the tubes of the rete testis. And then what's going to happen is you're going to have little tubes that connect tubes in the rete testis to portions of the epididymis along its length. Okay, and these tubes which move out of the testis and into the epididymis, these are known as efferent ductules. Okay, so you've got these efferent ductules which drain sperm out of the rete testis into the epididymis. Okay, so sperm which are produced in the seminiferous tubules, and the seminiferous tubules I've just drawn as a line rather than as a tube, but this represents tiny little tubes. Okay, so these in orange here, these are tiny little tubes. Okay, the sperm that are produced in the seminiferous tubules are going to drain into, they're going to move up the seminiferous tubules and then drain into tubes that are part of the rete testis, which is this great meshwork of tubes. Okay, then you're going to have tubes that leave the rete testis and drain sperm, from the rete testis into the epididymis at some point along the length of the epididymis. So remember, the epididymis is this wrapped around single tube. It's not a meshwork of tubes like the rete testis. It's just one long tube that's coiled around and around and around, and then is eventually continuous with the vas deferens, which is just a simple tube. Okay, right, so that's the vas deferens at the testicle side. Now what we want to see is the other side of the vas deferens. So if you follow this for long enough, you get to a point where you are behind the bladder, and this is what I'm going to show now. Okay, so this rises up from the scrotum, and then it enters into the abdominal cavity through the inguinal canal. Okay, and then it's going to end up behind the bladder. Okay, so shall I get a new piece of paper to show this? I think I shall. Okay, so let's start by showing the bladder then. Okay, so we'll put the bladder here. Okay, so the bladder will have the urethra coming off from the bottom here. Okay, so this is the, and I shouldn't be writing on there, I'm going to need to draw stuff behind it in a moment. Okay, so we're viewing from the front. This is the bladder here. Okay, this structure at the bottom, this is the urethra. This is where the urine is going to drain into. And then the urethra is going to go through, in males at least, and of course we're discussing this purely in males now. Okay, I should have probably at some point said that. I hope it's obvious. Okay, the urethra is going to go through the prostate gland, which is sitting underneath the bladder, and of course the prostate gland is only present in males. Okay, so this is the prostate gland here. Okay, and that portion of the urethra which goes through the prostate is known as the prostatic urethra. Okay, so I'll colour a few things in here. So we'll colour in the prostate in orange. So the prostate is a sort of little egg-shaped structure through which the urethra is going to pass through the middle. Okay, so this is the prostate gland with the prostatic urethra passing through the middle. Okay, right. Now, imagine we can see through the bladder and we can see what's on the back wall of the bladder. Basically, what happens is the vas deferens comes up behind the bladder like so, and this is the right vas deferens, and here is the left vas deferens, and then it's going to join with two glands that are on either side, sitting behind the bladder. Okay, and I'll put these here, and these glands are known as the seminal vesicles, and they contribute seminal fluid. Okay, like so. And when the vas deferens, and I'll just show this now for one of them. Okay, so when the vas deferens here, okay, joins with the seminal vesicle tube here, so this is the seminal vesicle, the tube that you then get is now called the ejaculatory duct, and the ejaculatory duct is going to move through the prostate and join onto the prostatic urethra. Okay, and I'm sorry it's smudged a little bit. That then there is the ejaculatory duct, and I'll colour code this in a minute. Okay, so that's the ejaculatory duct. So let's colour code these different portions then. So the ejaculatory duct is in green here, that's the ejaculatory duct. The vas deferens is this portion in red here. Okay. The seminal vesicle is all of this portion here. Okay, that drains into the vas deferens, and then together they form the ejaculatory duct. And then the same thing is going to happen on the other side. So again, you'll have the left ejaculatory duct here. Okay. So there are two separate ejaculatory ducts, a right ejaculatory duct and a left ejaculatory duct, and they're both joining onto the prostatic urethra separately by penetrating firstly through the back of the prostate. Okay, right, so there's a little bit of anatomy. That's put what the vas deferens is firmly in our heads. Okay, it is this tube that takes sperm from the testicles up to the prostate gland, basically, ready for them to be ejaculated into the urethra and then out of the urethra. Okay, so in people with cystic fibrosis, it is very, very common for the vas deferens to be absent on both sides, basically. So you have no left vas deferens and no right vas deferens. Okay, and what appears to happen is during development, the vas deferens forms, but then it degenerates, okay, in people with cystic fibrosis, and by the time they're born, they have no vas deferens, which is why it's congenital bilateral absence of the vas deferens, because it's from birth. That's what congenital means. Okay, so, why does this happen then? Okay, so, basically, again, the vas deferens is lubricated by a mucus, and the cells of the vas deferens appear to use the CFTR, the cystic fibrosis transmembrane conductance regulator, in order to make the serous secretion that then has the mucin added to it to create the mucus. And again, you get a very thick, sticky mucus clogging up the vas deferens during development, and that somehow causes them to deteriorate. The actual mechanism by which it actually causes them to degenerate isn't well understood, but that seems to be what happens, that a thick, sticky mucus afflicts the vas deferens as well, and it causes them to degenerate. Okay, now clearly this causes male infertility, because if you can't get sperm into the ejaculate, then the ejaculate will not be able to fertilize eggs. Okay, you will still have ejaculation because most of the ejaculate doesn't actually consist of sperm. It consists of the fluid from the seminal vesicles and also fluid from the prostate gland, but the ejaculate is not going to have any sperm in it, so it's not going to be able to fertilize an egg. And there's a fancy word for this. It's known as azoospermia. Okay, which means that your ejaculate doesn't contain any sperm. So azoospermia means that the ejaculate that you produce doesn't actually contain any sperm. Okay, and this is one of the things that if you have very mild mutations in CFTR, you get congenital bilateral absence of the vas deferens, even though you don't get some of the other symptoms of cystic fibrosis that we've discussed. So for instance, if you have 2 class 5 mutations in CFTR, okay, which remember were mutations which caused reduced production of CFTR by affecting the promoter region. Okay, so you have little CFTR, but it's much bigger than it would be if, for instance, you had 2 delta F508 mutations. Now, that might not be enough to cause the pulmonary problems, it might not be enough to cause the pancreatic problems or the intestinal problems. It'll cause the sweat problems. Your sweat will contain a higher amount of salt. But the other thing that it will commonly cause is, in males, congenital bilateral absence of the vas deferens. Okay, and often in people with these milder forms of cystic fibrosis, they won't even know they have cystic fibrosis until they then have problems producing a baby, okay, with conception. Okay, right. So, we will call it there for this video, and in the next video what we'll do is begin the discussion of the medications that are becoming available to treat cystic fibrosis.